Hey everyone, in this lecture we will be discussing cranial nerve number 1, the olfactory nerve. The olfactory nerve is a special visceral afferent nerve that courses from the cribriform plate of the ethmoid bone to the primary olfactory cortex found in the uncus of the temporal lobe. Here I'll be illustrating the mid-sagittal and coronal views of the ethmoid bone. Let's run through some quick anatomy pertinent to the video for this lecture. In the mid-sagittal view, here is the cribriform plate of the ethmoid bone. Here you have the perpendicular plate, mesoconchi, crista galli. Before anything can occur, there needs to be a stimulant. The stimulant in this case is an odor. It can be any odor. What I want you guys to do is think of your favorite odor and apply it to this video. Let's say that odor is within your surroundings. Eventually, that odor will travel up your nostrils, travel in between the perpendicular plate and mesoconchi of the ethmoid bone until it reaches a layer within the nasal epithelium named the olfactory mucosa. Within the olfactory mucosa, there are 4 special cells. Basal cells, sustentacular cells, olfactory receptor cells, and Bowman's glands. The basal cells are responsible for creating the stem cells from which new olfactory cells are born. Sustentacular cells are responsible for providing structural support to everything that lives in the olfactory mucosa. Bowman's glands are responsible for lubricating everything within the olfactory mucosa by secreting mucus. And the olfactory receptor cells are divided into 2 parts. The dendritic process and central process. As soon as the odor arrives at the olfactory mucosa, it will come in contact with the dendritic process of the olfactory receptor cell. Once that happens, a signal will be passed on into the cell and it will travel up past the central process of the olfactory receptor cell until it reaches the olfactory bulb. This structure is located in the anterior cranial fossa of the skull. Let's say this is the olfactory bulb. Within the olfactory bulb, there are two special projection neurons. They are the mitral cells and tufted cells. Once the signal arrives at the olfactory bulb, it will come in contact with both of these cells. As soon as they meet, they will form several synaptic glomerulus. Within each synaptic glomeruli, local microprocessing will occur. Thereafter, the signal will travel through the olfactory tract until it reaches the olfactory trigone. This structure here is the olfactory tubercle. This will enter the anterior perforated surface. Here you have both the olfactory medial striae and lateral striae. The medial striae will take the signal across the anterior commissure to meet with the signal on the contralateral side. This will allow us to experience autonomic responses that can come with the smell of food, such as increased salivation and gastric secretions. The lateral striae will take the signal to the uncus of the temporal lobe until it arrives at the primary olfactory cortex. And that is where the signal will finally be interpreted and the smell will be identified by our brain. And that, ladies and gentlemen, is cranial nerve number 1, the olfactory nerve.